Hello everyone. Today I will be talking to you about our research on alterations in retinal oxygen delivery and metabolism under retinal ischemia. As you know, adequate oxygen supplied by the retinal vasculature is necessary for cellular metabolism in order to maintain a normal retinal function. However, inadequate oxygen supply, or retinal ischemia, is implicated in pathologies associated with a number of ocular diseases. Therefore, it is important for us to gain knowledge of retinal oxygenation to better understand the pathophysiology of retinal ischemic conditions, with the ultimate goal of being able to advance therapeutic interventions for retinal hypoxia. Our study is based on the premise that a reduction in oxygen delivery causes retinal tissue hypoxia, and this can in turn impair oxygen metabolism and energy production, eventually leading to cell death and vision loss. The purpose of our study was to first establish impairments in oxygen delivery and metabolism in an experimental model of retinal ischemia reperfusion injury, and second, to relate the reduction in oxygen metabolism to visual function and retinal anatomical outcomes. The model of retinal ischemia reperfusion injury that was used in our study was a reversible ophthalmic vessel occlusion in rats. In one eye, we used a clamp to occlude the ophthalmic vessels to cause variable durations of ischemia between 60 and 180 minutes. Then we removed the clamp to resume blood flow to the eye and let reperfusion occur for durations of 1 hour or 7 days. We also did the same procedure in a sham group of animals in which duration of ischemia is specified by 0. A total of 61 rats were used in the study. This table gives the breakdown of how many animals we had in each of the groups. We performed multimodal imaging to measure vascular oxygen content, blood flow, and retinal thickness. Using our custom-built imaging system, phosphorescence lifetime imaging was performed following intravenous delivery of an oxygen-sensitive molecular probe. We used a frequency domain approach to measure the phosphorescence lifetime and convert it to oxygen tension values. For blood flow measurements, we used two imaging modalities. First, blood velocity was measured following intravenous delivery of fluorescence microspheres. A sequence of images acquired over time was analyzed to measure blood velocity. Second, we performed reflectance imaging. Retinal vessels were automatically segmented to determine the boundaries and calculate the vessel diameter. Finally, optical coherence tomography was performed using a commercially available instrument to measure total retinal thickness. For measuring visual function, we performed electroretinography ERG using a commercially available system. Here's an example of a phosphorescence optical section image in a rat retina. We optically separate structures at various retinal depths by projecting a laser on the retina at an angle. On the left panel, you can visualize the phosphorescence emission in retinal arteries, veins, and capillaries optically separated from the choroid. The middle panel shows measurements of oxygen tension in arteries and veins at one location on the retina. The right panel shows color-coded measurements of oxygen tension in all retinal arteries and veins. For each vessel, we calculate oxygen content. For each eye, we computed mean values for arterial and venous oxygen contents and calculated arterial-venous oxygen content difference. This slide shows our blood flow imaging protocol. On the left panel, you can visualize the motion of the microspheres within the retinal vasculature. The middle panel displays the locations of the tracked microspheres over time along a retinal vessel, which is used to determine blood velocity. The right panel shows a reflectance image with detected boundaries of a vessel outlined and used to measure vessel diameter. In each retinal vein, blood flow was calculated from the velocity and diameter measurements. A total retinal blood flow was then calculated by summing the flow in all the veins. Based on vascular oxygen content and blood flow measurements, we defined three oxygen metrics. First, inner retina oxygen delivery rate, DO2, as the product of blood flow and the retinal arterial oxygen content. Second, inner retina oxygen metabolism rate, MO2, as the product of blood flow times the arterial venous oxygen content difference. And third, oxygen extraction fraction, OEF, as the ratio of oxygen metabolism to delivery. The left panel here shows the location of the OCT raster scan temporal to the optic nerve head in a rat retina, and an example of one OCT image. The total retinal thickness was measured as the depth separation between the inner limiting membrane and RPE. The right panel is an example of an ERG waveform obtained in a rat retina. ERG B wave amplitude was determined from the waveform as shown. This slide shows the results of total retinal blood flow on the left panel and DO2 on the right panel as a function of ischemia duration and stratified by the two reperfusion durations. Data obtained after 1 hour of reperfusion is shown as blue bars, and after 7 days of reperfusion is shown as red bars. We found that after both reperfusion durations, blood flow was reduced as compared to the sham, only with ischemia durations of 180 minutes. Similarly, DO2 was reduced with 180 minutes of ischemia followed by 1 hour of reperfusion and recovered after 7 days of reperfusion. This slide shows the results of MO2 on the left panel and OEF on the right panel. Mean values are reported at different durations of ischemia and reperfusion. After 1 hour of reperfusion, MO2 was reduced with ischemia durations of 60 minutes or more. After 7 days of reperfusion, MO2 recovered only with 60 minutes of ischemia. OEF was also reduced with ischemia durations of 90 minutes or more, but showed recovery with ischemia durations of 120 minutes or less. Here is shown the results of the visual function on the left panel and retinal thickness on the right panel, plotted as a function of ischemia duration after 7 days of reperfusion. The ERG B wave amplitude was reduced with ischemia durations of 90 minutes or more, while retinal thinning was observed with ischemia durations of 120 minutes or more. Here are shown the results of correlations of MO2 with the two outcomes of visual function and retinal thickness after 7 days of reperfusion. Based on compiled data from all ischemia durations, decreased MO2 was correlated with ERG B wave amplitude reduction and also with retinal thinning. We were interested in investigating the relationships of MO2, B wave amplitude, and retinal thickness with ischemia duration. So as a first approximation, we assumed linear relationships. Fitting the data points provided regression lines with the following parameters of slopes and y-intercepts. There were significant linear relationships of all metrics with duration of ischemia. Next, we wanted to know whether outcomes, namely visual function and retinal thickness, can be predicted based on MO2 measured shortly after ischemia reperfusion injury. First, we defined an impairment index at each ischemia duration based on the calculated metric and y-intercept from the regression models. Note the impairment index has a range of 0–1 representing no impairment or maximum impairment respectively. The plot shows MO2 impairment index after 1 hour of reperfusion as a predictor for outcome impairment indices after 7 days of reperfusion. The data shows linear relationships such that from the equations of these lines we can estimate the level of impairment in visual function and retinal thickness outcomes based on MO2 measurements. Summarizing our results, we found that DO2 can recover after ischemia durations up to 180 minutes, while MO2 recovery was observed only after ischemia duration of 60 minutes. We found both MO2 and visual functions were reduced with ischemia durations of 90 minutes or more, while retinal thinning was detected with ischemia durations of 120 minutes or more. Third, MO2 impairment measured shortly after ischemia reperfusion injury was shown to predict visual function and retinal thickness outcomes. Finally, since MO2 can be measured in humans, future studies may establish prognostic value of MO2 in patients. I would like to acknowledge our funding support from NEI and Research to Prevent Blindness Foundation. Thank you for your attention.